Hello and welcome to this week's What Were They Thinking? We begin with the official naming of the new coronavirus. After careful consideration and attention to the feelings of various parties, the World Health Organization has decided to give the coronavirus that originated in Wuhan the name COVID-19. The name is an unimaginative abbreviation of CO for corona, VI for virus, and D for disease. The 19 designates what year it was first noticed in. In the words of one of the World Health Organization officials, the name was chosen to avoid reference to a specific geographical location, animal species, or group of people. They believe this will help in avoiding stigmatization. This news comes at a very bad time. It was thought that the disease had reached a state of plateau. Numbers were not drastically increasing, and there was at least limited hope that we might be able to limit its spread. This all changed when 14,000 new cases were announced in under a week. These new cases should be considered with a grain of salt. They are based on a combination of both genetic testing and scans of the respiratory system. Due to the scope of the disease's spread, there has been no way to conduct the in-depth testing required to prove, with genetics, that someone has the coronavirus. This has led to proxy examinations. These are not as good, but they do give at least some sort of positive response. This only adds to concern about how widespread, how virulent, and how dangerous it is. The disease has at least been seen in general terms as a looming threat, not a current threat. The rate at which it is spreading and the number of individuals dying from it are beginning to question that assumption. Although a scan may not be the best diagnostic method for identifying the virus, there have been efforts to visually identify the virus. Recently, microscopic images of the COVID-19 virus have been achieved. This was done using scanning and transmission electron microscopy. This allowed the National Institute of Allergy and Infectious Disease at the Rocky Mountains laboratories to produce a visual image of what the virus looks like. In the past, we've had far lower resolution images of the SARS virus to use, and in broad terms, there is a lot of similarity. Although visually very similar to the SARS virus and even the Middle East respiratory syndrome, the genetic differences make a world of difference. They are small and could realistically be counted on the digits of your hands and feet. They do have significant impacts on how virulent and how pathogenic the disease is. This news and others has unfortunately led to a lot of misinformation, primarily relating to the spread and degree of death the COVID-19 virus has caused. At present, it is estimated that over 1,000 people have died from the COVID-19 and more than 42,000 are infected. This is why it was declared a global health emergency. Some unscrupulous individuals are using the fear and paranoia to spread both misinformation about COVID-19 and how to treat it. Fortunately, there are a few tools available. Business Insider has produced a very good article summarizing some of these tools that they believe provide up-to-date information that is reliable. This is important. Information on the COVID-19 should be reliable. Countries like India have ignored this when they suggested individuals consume homeopathic medicine. others have tried to argue for the use of unproven and highly dangerous chemicals. Going now from the controversy of the coronavirus to some early experiments into immunology and mouse brains. A recent study questions whether or not it is just 10% of the neurons that are thought to be standbys in the brain. These were generally thought to be a form of structural element that kept all the cells where they should be. It's now believed that these same cells might play a part in amnesia, Alzheimer's, and other memory disorders after a publication in Science. These cells are known as microglia, and if they do play such a major part in memory retention, it could have huge implications for future treatments. The experiment underlying this argument used groups of mice who were conditioned to have a fear response to a specific environment. The microglia were then removed using a variety of techniques, whether that was pharmacology, biochemical, or genetic. They were then put back into the same environment to see whether or not they exhibited the same fear response. There was a drastic decrease in what the animals remembered and how they exhibited that fear response. This is unfortunately nowhere near definitive, as the brain is incredibly complex, both within mice and humans. As has been touched on previously, not all mouse brain studies can be translated to humans. There are also substantial issues with what kinds of drugs and chemicals were used, as these can often have unintended consequences, even at extremely low doses, that may further confound the experiment. In far more controversial and hopeful news, 3 US cancer patients have had their genes edited with the somewhat controversial technique known as CRISPR. CRISPR has come up before in relation to the CRISPR babies in China, who we now know are likely to have been a very badly designed and executed human trial, to put it in the best of light. The technique used here is far less invasive and dangerous. The researchers used T cells, an important part of the immune system that typically focuses on errant or rogue self cells. They then modified these to be able to better identify the cancer cells and put them back into the patients. The patient's immune system was able to utilize these cells to more effectively target and destroy the tumors. That would be the best case scenario. In this study, 2 of the patients had a stable condition established. This meant they were not going to get any worse for now. The 3rd patient had a more substantial response with a 50% decrease in the abdominal mass. This carried on for 4 months. Unfortunately, other lesions continued to grow. The cells themselves were stable genetically, which means the patient had them for at least 9 months after they were given their dose. This is a single dose of T cells. They were genetically stable, which is important for the long-term stability of this as a treatment and targeted treatment for a particular kind of cancer. Ultimately, 2 of the 3 patients have continued to survive. The 3rd did die from the progression of their cancer. This should make clear that CRISPR is no silver bullet for all medical conditions at present. In fact, it is at the moment only likely to be an unreliable and very early stage tool in the kit available to oncologists. If the only concern to oncology patients is that they have access to the best possible doctors and best possible treatment, the world would be a far better place. Unfortunately, that is not what is happening. In a recent announcement, doctors have had to warn people that they should not be inserting vegetables and fruits into their vagina. One of the newer fad options has been parsley. That leafy green thing you found spread across most foods in a restaurant has been suggested to help revitalize your body when inserted into your vagina, according to the Marie Claire. Part of it has said that parsley can help to soften the cervix and level out hormonal imbalances. Both of these sound like extremely bizarre claims if you understand anything about either anatomy or basic physiology. Inserting a foreign object into your vagina is unlikely to ever have that kind of influence on those hormones. It will, however, influence your stress hormones. These could well have an effect, but not the kind that you are after. That's the first and most obvious effect. But when you consider the details of what is described, that it should be fresh parsley and only a small amount, you need to worry about other complications. If you follow the suggestion of using organic parsley, this is only going to be more likely. The biggest issue is the introduction of foreign pathogens. Being a plant, and based on the fact that parsley can either be a leaf or root vegetable, you're likely to encounter E. Coli, possibly yeast, and other microorganisms that could be pathogenic. These microorganisms will hitch a ride right into where they shouldn't be and establish a home. This can lead to severe, long-term complications. This includes miscarriage and sterility. In the short term, you could develop a urinary tract infection. While speaking of fresh produce, there is an expectation that in the coming weeks, months, and years, we could see one of the most devastating locust plagues in recent history. Large parts of East Africa could be affected by the growing number of desert locusts that are occupying parts of Africa, in particular Somalia. Somalia is better known for its piracy, but it's also one of the regions that produces food for a large population, a population of about 10 million. They are already in a food crisis, and given the inaccessibility of Somalia, that could get worse. The volatility of the region means that aerial spraying to control the locust population is currently not being achieved. Over the coming 3 to 4 weeks, juvenile locusts could grow to maturity and develop wings. From here, they can travel to neighboring Kenya and Ethiopia. This means we could have 3 major countries on the eastern side of Africa experiencing massive food shortages. This presents another problem with the possibility of a second generation occurring in 1 year. The recent seasons and weather have contributed to the fast development of this generation. If the weather continues in a similar pattern to what is expected, then it is entirely possible a second generation could occur. The combination of moisture and heat will lead to reproduction and the locust spreading to yet more countries further abroad. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.